Okay, this is from chapter 3, which discusses organic molecules, and specifically we're in section 3.4. We'd already talked about the structure of proteins, that proteins are a polymer that are made up of amino acids. The amino acids are the monomers. When two amino acids are bonded together, it forms a dipeptide. The bond holding those amino acids together is called a peptide bond. Thus, anytime you see the term peptide, realize it has something to do with proteins and amino acids. Okay, so let's get up to this point right here. The structure of proteins is more involved than what we had when we talked about carbohydrates or talked about the lipids. And so, talk about the idea how if we would change in this chain of maybe 300, 400 amino acids, if we would change just one amino acid, it would change the final shape of that protein. And so the shape of the protein is critical to the protein being able to function or to function properly. And so when we look at proteins, we have a little bit more involved with the structure. So let's look at this. We've got 4 levels of protein structure. The first level is called the primary structure, and this is just the sequence of amino acids. So amino acid number 1, number 17, number 10, another copy of 10, another copy of 17, 2 copies of 16, so forth. And this forms a polypeptide chain, but it is not a protein. So primary structure, just the sequence of amino acids. Secondary structure, this is where this polypeptide chain will form a helix or a flat folded sheet. And this is due to hydrogen bonds because of the repeating amino acids. Okay, the third level of structure is called the tertiary structure. And this is where the helix or the sheet folds and bends into some three-dimensional shape. Now, within this three-dimensional shape, we're going to have all sorts of bonds. We're going to have hydrogen bonds, covalent bonds, disulfide bonds, and also hydrophobic interactions, which hydrophobic interactions, this is the result of water pushing these hydrophobic areas of the polypeptide chain away from the water, and thus pushing them in together, forcing them in together in order to exclude the water. And so all these different mechanisms are going to be involved in folding and bending this chain of amino acids into some three-dimensional shape. And so you can see if we would change one amino acid, let's take this disulfide bond. If we would drop out one of these sulfur-containing amino acids from the chain, all of a sudden that disulfide bond would not occur when this tertiary structure would form, and this chain of amino acids would not fold and bend the same way. So it wouldn't take on its proper shape, just changing one amino acid. Okay, now we've got a fourth level of structure. Well, hang on. Typically, a protein is going to form with the nonpolar hydrophobic amino acid R groups oriented to the inside of the protein. That is, they are pushed away from the water, and then the polar hydrophobic R groups are going to be attracted to the water, and they'll be oriented towards the outside of the protein. Okay, now that we have the polypeptide chain folded into some three-dimensional shape, it is actually a protein. So protein is going to have this right here, primary, secondary, and tertiary level of structure. Okay. Now then, all proteins will have those first 3 levels of structure. Some proteins will have a 4th level of structure called a quaternary structure, and this is where multiple polypeptide chains are intertwined or bonded together. So all proteins have the first three levels of structure. Some will have this fourth level of structure. So here's our diagram. This would be the primary structure, and we just have this chain of amino acids. Okay. Now then, go to the second level of structure, secondary structure. We get this hydrogen bonding, which produces either a helix, like here at the top, or this hydrogen bonding will produce flat folded sheets. Okay, so that's the second level of structure. Third level of structure, or tertiary structure, is where this helix, or the flat folded sheets, will get all these bonds across here that will link different parts of this chain and will cause it to fold and bend into some three-dimensional shape. And again, you notice the type of bonds here. There's hydrogen bonds, disulfide bonds, ionic bonds, and these hydrophobic interactions. Should be covalent bonds in there somewhere. I guess they just didn't make the diagram though. So all types of bonds. Now at this point that we have the tertiary structure, this chain of amino acids has a very specific three-dimensional shape to it, and it is now a protein. Okay. Now some proteins will have this fourth level of structure where we have several different polypeptide chains intertwined with each other. So right here in this hemoglobin molecule, we've got 1, 2, 3, 4 different polypeptide chains that are associated with each other to form this hemoglobin protein. Here's collagen, material that very tough material that kind of holds everything together in the body. And you have these collagen, these polypeptide chains that are all intertwined here to form this collagen protein. So some proteins will have this quaternary or fourth level of structure. All proteins will have the first, second, and third levels of structure, primary, secondary, and tertiary. Okay, disruption of protein shape. Well, protein shape could be disrupted due to the genetic makeup of the individual. The DNA is a set of instructions telling the cell how to assemble a protein. If you have a change in the DNA sequence within a gene, it's going to change the amino acid sequence, and this could possibly make the protein non-functional. So a lot of the genetic diseases, things like Tay-Sachs, sickle cell, albinism, are due to a person having the incorrect genetic code, and because they have the incorrect genetic code, it causes a critical protein to be made incorrectly, and that protein is non-functional. Okay, the other way that we could have a disruption of protein shape is kind of after the fact. Protein has been made correctly, but then some environmental factor disrupts the shape of the protein. So we have what's called denaturation, or to denature a protein is to take away its nature, to take away its characteristics. So to denature a protein is to change the protein's shape and cause a loss of function. And environmental factors that can denature protein are things such as high heat, extreme pH, high salts, high concentration of salts, certain chemicals like alcohol. These can all disrupt the shape of proteins and destroy or denature those proteins. So our examples here: fried egg. You drop an egg into a frying pan and it is a thick, viscous liquid. When it hits that frying pan, it very quickly changes to a solid. Those bonds in those polypeptide chains and those proteins, that bonds have been broken, new bonds have been formed, and it changes the nature of the material. It denatures the protein. Now, the Siamese cat, you might be wondering about that one. You know, a Siamese cat has a tan body, and then it has more pigment in the ears, the tail, and the paws. Well, the reason the reason for that is the core of the body is slightly warmer than the extremities. And one of the enzymes involved in producing the pigment in this cat is broken down by that slightly elevated temperature in the core of the body. So the animal has less pigment in the core of the body than it has out on the cooler extremities. That would be an example of just that slight temperature difference being enough to denature a protein. Okay, the last topic we want to look at is nucleic acids. Now the name tells you a lot right here, because nucleic acids are substances that are found in the nucleus of the cell, and guess what? They're acidic. Thus, nucleic acids. Okay. Some of the more common nucleic acids that we encounter are the genetic materials of DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. And again, I want you to be familiar with the example of polymer and monomer for all these different categories. Our polymer here is the nucleic acid. This is the DNA or RNA. The monomers are units called nucleotides. Okay, now the nucleotides have three components to them. They have a 5-carbon sugar, either ribose or deoxyribose sugar, a phosphate group, and a nitrogen-containing base. Okay, here's the diagram from your textbook showing this. So you've got phosphate, sugar, and a nitrogen-containing base. Now notice right here, here's a different version saying the same thing. Here's our nucleotide, it's a monomer. We've got a phosphate bonded to a sugar, bonded to a nitrogenous base. And so that would be our monomer. DNA or RNA would be our polymer. So we have, here's phosphate, sugar, base. That would be a nucleotide bonded to base, sugar, phosphate. There's another nucleotide. Go on down here, phosphate, sugar, base, another nucleotide. So the polymer is this long chain of nucleotides. And here's the specific nucleotides that we have in DNA. We have guanine is always bonded to cytosine. And then there's adenine, which is always bonded to thymine. Okay, then this is kind of a description of what we have in RNA and DNA and the functions of these nucleic acids. So let's just go on down here. Here's the diagrams from your textbook showing the different nitrogenous bases associated with the nucleotides. And then we have a few other nucleotides besides the genetic material that instead of forming polymers, they just act as one or two nucleotides, and they don't really form a nucleic acid or a polymer. And this would be ATP, adenosine triphosphate, and NAD, nicotinamide adenine dinucleotide. And we'll study both of these in a lot more detail when we get into the chapters dealing with energy transfer. Okay, and then at the end of your chapter, you have this summary that kind of summarizes, and I guess that's what a summary does. It summarizes the major details of the chapter with these different categories of carbohydrates, lipids, proteins, and nucleic acids. Okay, well that wraps up this chapter 3, and we'll go over this a bit more kind of as a review in class and get ready for an exam. Thank you. So long.